I want to honor your time, so I'm going to go ahead and introduce you, and then we'll get started. Thank you, Amber, for being here. Amber Salzman, PhD, is EpiCrispr's Chief Executive Officer and Director. Dr. Salzman is a leader with more than 30 years of experience in the pharmaceutical industry. Before joining EpiCrispr, Dr. Salzman served as the President and CEO of Ohana Biosciences. Dr. Salzman currently serves on the Osler Diagnostics and the Avideo Bio boards, both in the UK. Dr. Salzman received her bachelor's degree from Temple University and holds a PhD in mathematics from Bryn Mawr College. In addition to advocating for patients living with rare diseases, Dr. Salzman leads the Stop ALD Foundation, a nonprofit medical research foundation focused on developing novel gene therapies for ALD. Thank you, Dr. Salzman, for giving us your time. I know you're very busy, and I want to commend you. You are the first CEO of industry to ever join the parents group. So thank you so much for joining us. In fact, I think you're the first one from industry, period. So we appreciate your time and your transparency. And I will let you take it. I just want to
Sure.
remind folks, please mute your mics when you're not talking. And Dr. Salzman will take your questions as she's giving her presentation. So either raise your hand or jump in or put it in the chat, and then we'll get to it. So thank you very much for being here, Dr. Salzman. It's a pleasure.
And please call me Amber. I will say that when I got my PhD, I did come home and told my whole family to call me Dr. Salzman. But I got over it now, so you can call me Amber.
Okay, thank you.
And thank you, Ellie, for inviting me. I mean, to me, this is a privilege. This is like why I love what I'm doing and this kind of engagement. So I really appreciate your time. I know it's the evening for everyone, so please do stop me with questions. So just to add to my background to give you a sense of why I do this. So I actually was at GlaxoSmithKline for 25 years and was heavily involved in drug development for all kinds of different indications, but they were mostly mainstream indications. And I was so excited when we would get to talk to patients and they would talk about what an impact. I mean, one of the stories that always comes to mind is after 9/11, we did a lot of respiratory stuff. After 9/11, we got a letter from a patient saying, thank you. Without Advair, I wouldn't have made it out of the towers because I would have, they had asthma. So it's that type of stuff that would keep me going. But I never thought I would be the patient on the other side. So then while I was at GSK, about almost 25 years ago, my oldest nephew was diagnosed with adrenoleukodystrophy. And as you know, when you find a genetic disease in the family, it's kind of a family affair party and everybody gets to join. So we tested all the members and my son tested positive. He was one at the time, as did another nephew. And unfortunately, my nephew Oliver, who was first diagnosed, it was too late. He passed when he was 12 years old. And at the time that my son got his diagnosis, he had like 3 or 4 years before the time bomb went off in his head. And all of a sudden, I realized there's all these diseases that really were not getting the attention it needed. I mean, academics were looking at it, but they were doing more research than saying we've got to find a cure for them. So while I was at GSK, and I was very fortunate leading development there because I would travel around the world for my job, so that was my day job. And at night, I would do all this work to try to find a cure for ALD. And ended up working along with academics and industry and all kinds of partnership to get a gene therapy ALD trial off the ground, which then a biotech took forward, and it took 20 years to get Skysona approved, but we finally got a treatment for ALD. And I thought, there's no way in hell anybody should have to wait 20 years. So I really that's what drove me into going into genetic medicine. And that's what I've been doing. I left GSK in 2008 and been leading private and public genetic companies since then to try to move things along much faster than 20 years. What pulled me into EpiCrispr, so while I did a lot for ALD, it turns out that my husband's family has FSHD. Now, when I first met my in-laws, I always noticed they had kind of a gait disturbance, but they didn't know what they had at the time. I've been married 35 years, so this goes back, as you know, a lot of the work is more recent. And then when they finally got a diagnosis, Catherine Wagner was my family's physician. I'm sure a lot of you know Catherine. Um, my family said, well, if you found a cure, if you work on a cure for ALD, what about FSHD? And I said, look, I'm not a bench scientist. I don't know what to target. I'm a drug developer. You give me a target, I will bust doors down and find a therapy. But if I don't know the target, I'm not equipped to do that. So then fast forward, somebody asked me if I would talk to EpiCrispr. And at the time, I was like, I don't really want a job on the West Coast. I'm an East Coast person. But someone said, please, you have to talk to them. And then I met the team there. And for those of you who met Alex Collins, she was there at the time. She's just so driven for patients. And she started telling me about how the platform that EpiCrispr had could address FSHD. And I was like, what? FSHD? You can cure FSHD? And then she explained to me how the technology got around all the limitations of other existing technologies. And I kept pushing, saying, well, what about this, what about that? And she's like, this is what I think the answer is, but we haven't done the work yet. And that's what drove me to take the job at EpiCrispr. So I joined EpiCrispr at the end of 2021. And at the time, the company wasn't working on FSHD, but we pulled together a proposal for the board of directors at the end of, I came in October. By December, I said, this should be our lead indication. We got to drive things forward. And I have to say, the FSHD Society really helped because we reached out. I'm sure some of you know Jamshid, who was there at the time. He helped us figure out what we should be looking at, where we should be going to get the patient cell lines. And I will say, the infrastructure that existed at that time really was what enabled us to go from an idea to having a clinical trial in 3.5 years. Like the infrastructure in terms of what patients and the experts had done is really, and also willing to work with us and educate us, review our data and see that it made sense and we weren't kidding ourselves. So that really, I will say, there was no way we could do it if we didn't completely leverage the community. And I say that because you guys are obviously very involved with trying to bring treatments forward. There's no way we could have ever moved forward that way without that kind of an infrastructure. So that's that really initiated taking things forward. We are leveraging, and I have slides, but I'll only bring them up if you want me to when you have questions. I think it's easier to talk through. I'm sure everyone's heard about CRISPR in the press, but CRISPR, as you know, basically is designed to bind to a location that needs to be corrected and cut out the bad DNA. For FSHD, we don't need to cut out DNA. That could cause a lot of problems. What we really need to do is go to that region, the D4Z4 region, which I'm sure you guys know about, which is hypomethylated and methylated. The tools that were available just couldn't do it. I mean, there's something called a Cas9 molecule, for example, which is what most people use. There's a couple of key real challenges with it. One, it's too big to fit in an AV vector. Two, it's bacterial, so you'd have an immune response to it, so you would clear it right away. So we had to come up with our own Cas molecules. But what I loved on the team at EpiCrispr, these were people, instead of saying, here's why it won't work, it was like, here's what we could do to make it work. And it really unbelievable, very strategic. And as you know, for a new modality, you got to go through a hell of a lot of testing. And also for the FDA to clear you going into patients. I mean, our just preclinical part of our submission to the FDA was thousands and thousands of pages. Our investigator brochures is hundreds of pages so that the physicians could really get a look at overall, do they feel comfortable even recommending to a patient to be on it? But we had to design our own Cas molecules, our own suppressors to suppress DUX4, and design our own guides, and then test it thoroughly in vitro, in mice. And the mice we used, because I know some of you have seen different mice models, we use a really challenging one, which comes from UMass, which was the xenograft. So you take a mouse, you take out their TA muscle with a toxin, and then when you have that niche in their muscle, you put an FSHD cell, so it grows and populates it. So that muscle is a human muscle growing. So that's what we tested all of our in vivo in. So we would make sure that we had a dose that do a dose response, make sure that we had a dose that would be efficacious. Then we had to deliver it. And this is where you probably have questions, so please stop me. In order to deliver such a construct to the muscle with today's tools, the best way we saw to do it that had the most experience was using the same vector that Sarepta uses for DMD, AVRH74. They've dosed about 1,000 patients, but I'm sure you've heard they've had some tragic events this year. They had a couple of deaths in the non-ambulatory patients, and then they had a death in their limb-girdle non-ambulatory adult patient. So I know a lot of people would say, walk away, RH74, no good, it kills people. We took the approach. Now we started working on it before the deaths, but we knew that there was, we ran a huge amount of tox studies, 6 months in monkeys and looking at all different things. And we really got a handle on when do you run into trouble. And just to add to that, our chief medical officer, Wes Miller, you all have to meet him. I just love working with him. Before he worked with us, one of the companies he worked with was Audentis, which Astellas purchased. They had a product for myotubular myopathy, which is really, really bad. I think the kids die before like 5, 6. And unfortunately, when they treated with an AAV, some of the kids did die. But he was he said he was hired like a week after he got there, the death started. So he had to really look into it and make sure that he could understand what was going on and, really get his arms around it. So he comes to us from, in some ways, the best experience possible because he had to dig into it and see how you get into trouble when you do high dose AAV. So he studied inside and out. Also because of his connections, people did share confidential data about the Elevidys deaths, etc., just to make sure that we knew confidently what the differences are. Now, and I'm happy to go into them if anybody wanted to. But we took the approach of don't let failures stop future successes. Learn from the failures, figure out what you got to do, because if you hit a brick wall, we say plow through it. Unless there's a way to veer right or veer left, you got to plow through it and figure out how to make it work. So that's really, we spent a lot of time testing it and the FDA wanted, look, I've been doing drug development for a long time. This was probably the most thorough and robust safety package I've ever submitted. Because it was new, they wanted to make sure we didn't have any off target. They wanted to make sure we had long-term studies. And then when it came to which centers we should run it at, we wanted to make sure that we partnered with physicians who knew a lot about FSHD, but also knew a lot about AAV. Fortunately, there are such physicians as part of the FSHD Clinical Trial Research Network. So part of the criteria was we wanted physicians who knew FSHD, who knew AAV, but we also wanted them distributed geographically so that it would be open to, or when I say open, at least convenient as possible to people in the US. So we have 5 sites. 3 of the 5 sites are part of the Clinical Trial Research Network. The other one, surprisingly, UMass, which so much comes out of their FSHD, is not part of the Clinical Trial Research Network. I have no idea why. And then there's another one, which is the Atlanta Rare Disease Center, which they have so much experience with this. And then, of course, we're using a very prominent FSHD site in New Zealand and Australia. So we were really fortunate that we had very, very experienced physicians who wanted to work with us, and they were excited about it. So I guess I want to stop talking because I'm so passionate about what I'm doing that I could keep you up all night, but I just want to pause and see if there's questions. Otherwise, I'll keep talking.
So I have a question about the AAV, and so you're not using a Cas9, you're using a Cas Onyx, right? And so I realize the AAV is still dangerous, but is it different with the non-bacterial? Does that affect it at all, or still is just with the danger with the AAV?
So yeah, let me separate the two because the Casonics is inside what the AV delivers. And Casonics, because it's non-bacterial, when it gets delivered, it doesn't evoke an immune response the way Cas9 does. So when you use AAV to deliver Cas9, there's data published on this that it does evoke an immune response, which can get you into some trouble. And can also be your body may get rid of it because it's got an immune response to it.
Right.
Because our Cas molecule is non-bacterial, we've done a huge amount of testing, and it doesn't evoke any immune response by humans. So that's good. As far as the AAV, you do high dose AAV, it's dangerous. I mean, anybody that tells you AAV is not dangerous is not being transparent. So I kind of liken it to chemotherapy. We know chemotherapy is dangerous, but we also know that it saves lives. So when we actually, oh, what was it? Few months ago, it was after the death that had, it was after at least one of the deaths at Sarepta, when we were trying to raise more money to make sure that we could go all the way. One of the investors said, are physicians and patients really going to use gene therapy for FSHD? FSHD isn't a bad disease, which I, of course, freaked out at because of my family. I'm like, in some ways, I hate to say it, and you guys know this, sometimes it's like an invisible one, because if you're just sitting, nobody notices any problem. And sometimes it frustrates me when I hear that, oh, it's slowly progressing, and I'm like, really? It's impacted my family. I mean, so my husband, one of my husband's cousins who has it, I mean, his wife is tiny, and when I see her trying to lift her heart, I mean, it's just like to say it doesn't impact them. I don't even. So we, as a result, we hired a third party to do interviews with patients and physicians. And I was really pleased that the physicians were like, no, this is a tool, FSHD warrants going this kind of route. So when I say it's not safe, I mean, obviously I wouldn't do something that we didn't think the risk benefit was there. But anybody that tells you that AAV at any dose is safe, like it's a medicine. I don't know any medicine that is completely safe. It's just you're always looking at the risk benefit profile. Now, I will say with AAV, we know that people's livers are particularly sensitive to it. So when I've had people ask me if they should go on the trial, I said, I'm not a physician and I am clearly not your physician. Here's who I would talk to, but I just want to, the one caution I would give you, you want a squeaky clean liver. You don't want to have any kind of predisposition. And just as a tangent on that, the problem that the Duchenne ran into a bit was that when you look at typical liver lab results, for those of you, I'm sure you guys have looked at your lab results or your kids, you look at ALT and AST. And if those are not normal, it usually signals that something could be going on in your liver, and then you do further testing. The challenge with Duchenne patients is their ALT and AST is always elevated, and the theory, or when I say theory, people who know a lot more than me say that that's because of the muscle breakdown. It leads to an elevation of ALT and AST. I won't tell you more. That's why they say it doesn't mean their liver's not healthy. So, but the challenge is when you're screening patients for Elevidys, for Sarepta's AAV drug, it could potentially mask if you really did have a liver problem. So physicians do look at other labs and other things, but what really gave us some comfort is that FSHD patients, you really should see normal ALT and AST levels because they should have very healthy livers. So I don't mean to ramble too much, but look, AAV, you wouldn't use it unless you really needed to. And that's why you have to use it cautiously and judiciously. And like I said, there's been 1,000 patients on it. All the ambulating patients, I mean, some of them have had their liver numbers go up, they've been managed and taken care of. Um, there's other reasons, which if you want, I can go into why the non-ambulatory ran into trouble. But it's a risk. I mean, I wouldn't be transparent if I didn't think so. But I always think, would I take it myself? I mean, I always personally, or would I give it to my child? Would I give it to, a relative? And that's kind of what I think about it. So yeah, it's a discussion to have with your physician based on your particular situation and what the risks are.
Well, you're definitely preaching to the choir here because most of our parents are children who are very severely affected. So we know how rapidly and how devastating
Yeah.
the disease can be. So we appreciate you saying that and being the champion out there to say that, because a lot of times people do just kind of frame it as a slow progressing kind of benign disease, and we know better. So thank you for that. I did have a question though, back to the bacterial, the non-bacterial with, I know there was a paper out there or something, again, I'm not a scientist, that that it washes out like the treatment, for lack of a better word, washes out. It being non-bacterial and not invoking that immune response, does it make
E-
it more durable?
Exactly, because your body tries to get rid of stuff that it doesn't want. So when it sees bacteria, it tries to clear it. So
Right.
that's why the paper has shown, and we've compared stuff head to head with Cas9. So in those papers, they showed that when they were using Cas9, it wasn't durable. Like it first got in, and then your body got rid of it. We've done 6-month monkey and mouse studies, and we've shown that the level of Epi-321 that you have at dosing is exactly the same level that you have 6 months later. I mean, it doesn't degrade at all over 6 months.
That's significant, isn't it?
Yeah, that's further reinforcement. So while we made sure in immunogenicity studies that humans did not respond to cationics, because we were worried that if they did, potentially they would clear it. But we just also wanted to, this is old belts and suspenders left and right. So we looked at monkeys 6 months out, and 6 months out, they had the exact same level that they had like shortly after dosing.
Nice. So the clinical trial that you're running right now, you've dosed with two patients, is that correct? Two
Yeah,
patients, one in the United States and one in New Zealand? Is that
yes.
right? Yeah. And you'll have data on them. Can you kind of walk us through like what that
Yeah,
kind of clinical trial process, you have to do some
the
depression?
pressure. Yeah,
Yeah,
yeah.
suppression. Okay.
Yeah, so first in terms of immunosuppression, then I'll tell you the cadence of the trial. So as I mentioned, your body does react to AAV. While it may not react to the payload, which is the Cas9 and I'm not Cas9, sorry, the Casonics and our specific construct, your body does respond to AAV. So a way to dampen that response is to immunosuppress. So patients are pre-treated with corticosteroids, which does open them up. I mean, you are somewhat immunosuppressed, so we recommend that people aren't going out in crowds and, potentially picking up an opportune cold or something. So you do start with corticosteroids. Then you have your one-time infusion. The trial is, oh God, I need Wes on the call. I think it's one night in the hospital because we want to make sure you're monitored, but then you have to stay nearby. I said there's 5 sites in the country, so you shouldn't have to travel too far, but you have to be close to the hospital because I think it's the first week you're in every day, just checking and monitoring and making sure. And then I don't remember the exact protocol. I should have brought it with me, but you're really monitored closely, then, less over time, and you wean off of your steroids after some time so that your body gets used to uh, handling it, and then you're okay. And there's, as you know, thousands and thousands of patients who have done this.
Mhm.
Um, so that now, in terms of the trial, so we were really pleased that the FDA, all right, so when you're doing gene therapy, because it takes time to see an adverse event, they usually require, the minimum we've ever seen is a month between dosing one patient to the next. We were really pleased to see that for EP321, even though it was a completely new modality, they were comfortable separating them by a month because people were worried they were going to separate it by 3 months, which would make this trial kind of tough to do. So basically, a month after the first patient is dosed, all of their labs, all of their data, and everything is reviewed by a safety monitoring committee. And there are an external bunch of physicians who look at it. They were, approved way before the trial started. They look at all of the labs and everything and say, yeah, this patient looks fine. Therefore, we think you're fine dosing the second patient. So that's what happens between each patient. And then after the third patient, which is scheduled, I don't want to jinx myself saying where and when, but we do have the third patient lined up. Things always happen. I mean, you catch a cold or whatever. After that person, that third patient has a month data, then all the three patients' data is pulled together. And if everything looks good, then we will up the dose a little bit higher. Now, having said that, our preclinical data, and we have extensive preclinical data, gives us reason to believe that we should have efficacy at the lower dose. But as you know, sometimes more is better. So that's why if everything looks good and the safety monitoring committee agrees, we will increase the dose. And then we still have to do that cadence of a month between each patient. After those three patients are dosed at that high dose, if everything is fine, then we don't have to wait anymore. Then we can dose the next three all the same day if we wanted to. Now, in terms of when when we'll really have data. So the safety monitoring committee is looking at, we're just throwing all the data at them. I mean, obviously we're organizing it, but like every little lab, everything. But everybody wants to know, well, how do we know if it's working or not? So, part of the way the study is designed, you get Epi-321, you take a muscle, you come back and you do a muscle biopsy, and believe me, I know how painful that is. You do a muscle biopsy at baseline, you do a muscle biopsy at 3 months and 12 months. So let me just talk about the muscle biopsy before I tell you about all the other stuff you do. So the muscle biopsy at 3 months will be compared to the muscle biopsy at 1 month to see all kinds of things, like did we actually increase methylation from baseline? And as we know, you don't have to increase it by much to basically stop symptoms. Did we increase methylation? How much of the vector got into each cell? Could we suppress DUX4 or any of the downstream markers of DUX4? So the muscle biopsy, I mean, and some of those things that I just mentioned are really hard when you're talking about a little tiny muscle biopsy, but we'll be looking at everything we can from that. So given the cadence of all of that, it takes about 2 months to do that analysis. So the first 3 patients, 3-month biopsies should be analyzed by first quarter next year. So by first quarter next year, we should have a sense of, did we in fact do what we thought we were doing when we delivered it? And then it's a year-long study. And then we're also at 6 months, we're doing MRIs, all the functional measures like 10-meter walk, run, reachable workspace, myometry testing, and things like that. Wait, I think somebody posted a.
Yeah, Debbie wants to know about ambulation status. If the liver's healthy, why would ambulation status matter?
Oh,
I hate, like, they talk about those who are non-ambulatory, and I feel like that's, in my view, unfairly blaming ambulation status. And I don't want for those in our community who are non-ambulatory to sort of, to have a
Okay.
problem. Yeah.
Thank you, Debbie. I agree. Okay. So, all right. Since you asked, I'll go into the tangent. So
Sorry, I didn't mean to
No,
stop your
no.
train of
I
thought
think
there. I
this
just
is
wanted to
really,
make sure we talked about it
I
at
think
some
it's
point.
really important. And especially when we talk about the fact that right now our study is only enrolling ambulating patients, and we want to expand it so that everybody could get it, kids as well. So here's there's other challenges I'll get to, but in terms of the AV and Sarepta, the non-ambulating patients suffer from a lot of muscle loss. So they're very sarcopenic. The way the doses are calculated is based on body mass, not muscle mass. So if the drug doesn't have a lot of muscle to get to, it's going to go to the liver. So you've totally overloaded the liver. Also, fat is not vascular. So it's like, it's not getting, fat's not getting any of this vector. So everything I'm saying is speculation. We have anecdotal data to support that. We actually want to do a study to show that you should be dosing based on muscle mass, not based on body weight, because if you dose it on body weight, you can get into trouble because you really overload the liver. So, and when I say anecdotes, for example, when we did our monkey studies, it's fine. We have a great preclinical guy. He used to be veterinarian, and he's like, when we do our monkey studies, I got to look these monkeys in the eye before we give them the drug. He wants to know, he's like, this one has alopecia. I don't know. Like, he really looked at everything. And he said, interestingly, the fat monkey had elevated ALT and AST. I mean, he was fine, but if you asked which one didn't do as well as the other, it was the fat monkey. So anecdotal, total anecdotal. And then I've talked to some academics in the field who have done a lot more than me. And I'm like, why aren't you guys studying this? He's like, well, studied in rodents, and that's true. You have to dose based on muscle mass, but every sponsor has got to do it on their own. So we designed a protocol. I just actually emailed Cure Duchenne last night to see if they could fund, we're just short on funds. If they could fund it, we're going to reach out to the MD. We're trying to figure out if somebody can help support that study because we want to do a real legit. I don't know why Sarepta didn't do it, to be honest. I'll have to ask them if there's any opportunity. I mean, they're so heads down. Last thing they need is me bothering them. But we want to do a real good study really
Mhm.
showing the difference when you're dosing based on muscle mass versus body weight, knowing that the vector doesn't go to fat, it goes to muscle. And if you don't have enough muscle to soak it in, it's going to the liver. Debbie, this
Mhm.
is
Okay.
totally not verified yet, but we've been hearing that from others as well. So I think that study
Yeah.
needs to be done.
Okay, so I just want to say this is a perfect example of why I appreciate what you're doing because we, I think that one of the frustrations has been that we've seen people sometimes want to do things the way they've been done before or think that that must be done. And we're just talking about a challenge, like the worst challenge is, having a death from a clinical trial. And instead of saying like, okay, well, we've got to exclude non-ambulatory people, let's figure out a solution. So I want to thank you for that because I feel like you have a lot of outside the box thinking, which maybe this disease needs. And I just one more thing I wanted to mention is that unfortunately, with this disease, some people, as you said, progress quickly. And for some people, it can be life-threatening. And there are some of us on the call who understand that there are risks, and we know what will happen to our children if nothing is done, or if we wait a very long time. So I just want to thank you for understanding that.
No, thanks for that, Debbie. And part of it is like I had to make these decisions for my son.
Yeah, yeah. So you get it. You really
And
understand.
what's really interesting, I mean, he's 25 now, and I guess a year or so ago, I had a conversation with him talking about some of the decisions I made. And he actually, I said, you know what, we could have waited. But he's like, thank you. He goes, thank you for making that decision. I was too young to make it, but I appreciate that you made the decision. I said, we were gambling with your life, and we had to live with that decision. And he said, I'm glad you did because he also understood what the consequences of not doing anything would
That's what I
be.
say. It's a gamble if you do nothing, or like you really know what's going to happen for some of these diseases if you do absolutely nothing.
And I hate to say this, but so the gene therapy wasn't ready in time for my son. So he had a bone marrow transplant. So which is the only other way to stop the disease. So we lived on the transplant unit for quite a while when he was recovering. So you get very close because you're in a HEPA filter situation, like you kind of are stuck in the family, not a place that anybody wants to be. So you got to know the families quite well. And unfortunately, probably about half the kids on the unit died while we were there. And when I would talk to those parents, the one thing I walked away with, which I really took as I moved forward, I mean, this is 23 years ago, the parents who felt like they did everything possible, even though they lost their child, which was horrible. It wasn't as horrible as those that felt they just could have tried going under another stone or something. Look, I don't know, everyone's different, but unfortunately, I was there for a lot of these horrible events. And nobody wants these kind of decisions, as we know. But they said they can live with the fact that they tried everything. I mean, they were leukemia kids as well as metabolic disorders and rare disease. It was about half and half for both. The family in particular we got close to was a boy with leukemia, and they were told that their odds were really slim. And they're like, no, we want to try everything. But yeah, look, like I said, I grew up at GSK thinking, oh, I'm helping making medicine. And then all of a sudden I ended up on the other side, and it's just a really different perspective.
Mhm.
It's a really
Well,
different perspective.
I think that's a good segue into going into other populations. I know you just started a trial. I know you have to get the data, but and
Yeah.
we've had some conversations. So what are you thinking about? I mean, do you want to share that with us about
Sure.
going
Yes.
to the
Look,
kids and going into maybe non-ambulatory? How do
I'm
you see that progressing?
I will be very transparent. So when we designed the trial, of course, we wanted to go after and help all patients. But we needed to be able to find a way to show whether the drug worked or not. And in gene therapy, it's open label, so you're not doing a placebo arm, you're just comparing it to natural history. So we had access to the Resolve data, which my understanding is there is a paper in that there's a lot of physicians working on it, but that they will publish it. I keep harassing Jeff every time I see him. But the beautiful part is they made the data available, and we had a statistician analyze it to say, if you looked at patients who had a little bit of symptoms but weren't too bad, how close was their natural history? If you expanded it, if you narrowed it. And where it ended up was we were able to say that if you had, Rishi score, some way to calculate the severity, a Rishi score between 2 and 4, then you could be somewhat predictive of what you would expect their decline to be in things like the 10-meter walk run. You would lose about 8% a year, or 7-8%. In terms of the quantitative myometry testing, you would lose more than 20% a year. So that if we stabilized patients, we could say, yeah, that wouldn't have happened without the drug. The challenge is, of course, if you're not within that controlled Rishi 2 to 4, you could be progressing either much slower or much faster. So it's hard to know whether the drug's working or not. So that's one challenge. So that's what explained how we picked the patients for this study. So if this looks good, the question is, how do we move on to the rest of the population? So there's two things. One, to dose kids, we need to prove to the FDA and ourselves for ethical reasons that it's reasonably safe and that there's a reason to believe it would work. Otherwise, you can't dose kids in a disease that also has adults. So assuming we get there, how do we then measure whether it works in kids or not? So we're trying all different ways. Allie's been great. I've been trying to figure out how we can get more natural history study on kids to say this is how they progress. And as you know, the big challenge with kids is, especially when you talk about a toddler versus a teenager, a toddler's growing muscle while they're losing muscle. It's much more challenging. So what we're trying to do is other type of exploratory stuff. So we're working with Springbok Analytics. You probably know they're the people that do whole body imaging. And they think that, at least with adults, they're able to progress based on your baseline whole body images, looking at how much fat fraction on your muscles and stuff like that, where you would end up. When I communicated with Ian, why am I
Woodcock.
Woodcock in Australia, he said they have scans from 50 kids. So he just needs some money for Springbok to add that to their analysis. So we're trying to push ways to get, more ways to measure that we're having an impact. The other thing we're trying to do, as you know, there's studies that correlate your methylation status with disease severity. So the less methylated, the more severe your disease is. And as you know, even with siblings that have the exact same mutations, one sibling who doesn't have symptoms, it's because they have more methylation. So when we met with the FDA, for example, and said, well, if we methylate that region, wouldn't you agree that we've had a positive impact? Because of, there's hundreds of patients that have shown disease severity correlates with methylation. And in the nicest way, they were kind of like, nice try. They're like, yeah, we see the correlation, but nobody has proven that if you change methylation, it changes the course of the disease. So we want to use this first study. It may not be enough patients. We may have to add some to prove that if you change methylation, it does in fact predict that you're going to change the course of the disease. If we can get there, then I don't have to get all crazy about this natural history because then we can just say, look, if we increase methylation, that's going to make a difference. So that's just to give you a sense of what we're trying to do to figure out how to expand it to all situations.
So you would use methylation as the biomarker, right? So if you could prove that methylation affects capability, right? Or alters disease progression, then based on that biomarker, you could then, without all the 10 meter walk, run, all that stuff, you could say, then I could go down and say kids or the non-ambulatory. And as long as I'm increasing methylation, that's the biomarker. But then would you still, for the FDA, have to prove like daily task improvement or functional improvement, or no?
So we don't, so much of this is before we see the data, if we had sufficient data to show them that increase in methylation is predictive of improvements in 10-meter walk, all that stuff, then we shouldn't have to do it. We probably would do it just to keep track of stuff, but we don't know what it would mean, because if you take a kid who's progressing rapidly and maybe you just improved it, but you didn't stop it, you may not be able to detect it, just compared to somebody who's progressing slowly. So we will absolutely measure everything that's reasonable to measure, even if the FDA said that, well, methylation is a good enough endpoint.
But that's a good place to extrapolate it from, to say, okay, this is it. So, and the Springbok data, we're working really hard to get this data from Ian.
Yeah.
And to get the MRI, and that will help as well, because then maybe you don't have to do biopsies. Is that right? Or will you always have to do a biopsy? Could you take, I know you talk a lot about digital twins. Could you take those MRI, Springbok MRI, and compare it to a child without doing like a biopsy?
Ellie, I really hope we get there. I mean, I've only had not the full biopsy. It hurts like hell. So yeah, I don't think we're going to get there right away, but I do think we will get there. The other thing is, there's the whole placebo arm from Fulcrum that Springbok was trying to load in to even improve further their machine learning. I actually sent a note earlier today to find out what the status of that was, because the more, the more data, the better their predictions are. That's a great question about the muscle biopsies. I think right now people would be nervous not to do it because that tells us so much right now. It tells us DUX4. Well, the other thing, Allie, as you know, Avidity had that circulating biomarker. We all want a circulating biomarker, in which case then you don't have to do these muscle biopsies. So that's another thing, either the imaging or a circulating biomarker. So because we can't gain access to the Avidity assay, we're working with Solve FSHD and the FSHD Society to figure out how we can come up with that circulating biomarker so everybody else can use it, so we can all compare head to head how that works. But that will be another way that we can hopefully start to move away from muscles. Yeah, that's so people are trying. I just don't know how soon we'll get there. I know these are really painful.
So I want to introduce you to Vera. Vera Trayance, she's in New Jersey. She's on the call. She has two little girls, 7 and 9, right, Vera? And they're
Yes, 7 and 9.
very severely affected. So I just wanted to introduce you to her. And Vera, do you have anything that you would like to say to Amber? Introduce yourself and talk about your girls a little bit, and if you have any questions.
Sure. So yeah, we were diagnosed 4 years ago. I went my whole life, 35. I had FSHD. Well, I have FSHD. So yeah, we're really, hopeful to see what EpiCrispr is doing. We're really happy to see that you have the rare pediatric disease designation. So, we're like you said, the benefit outweighs the risk. So, like in our situation, they're 16 months apart. So Isla's 9 years old, Sienna's 7. So, I see what's going to happen in 16 months. Isla just in the last month or so is now like a full-time wheelchair user. So, I'm hoping there will be an intervention before Sienna is, a full wheel, so I feel like we have 17 months for my younger daughter. So yeah,
And their
just,
methylation is very low, correct, Vera?
but yeah, so is mine. I have the same repeats and the same methylation as my daughter's.
Oh, so you
And
did actually get the methylation tested. That's good.
yeah, with Dr. Jones and yeah, the blood tests, yeah. Yeah, so yeah, I don't know if any like timeline you can give, anything we can do, to like
I mean, as you can imagine, we are pushing as fast as possible. I mean, I gotta tell you, I'm
Mhm.
surrounded by a team that literally, I mean, we're constantly sucking each other trying to help move things forward, but responsibly. I wish I could, I mean, like I said, even dosing patients right now, we have to wait a month between patients. So, I mean, I mean, what was interesting, we really liked that the second one was in New Zealand because it was Monday for us, Labor Day, but they, of course, were open on Monday. So, like we're doing anything to try to, for us, even a day, I'm like, patients are waiting. Every day is capabilities that you're losing.
So you
So,
said the first quarter that will be public, the data? It
yeah,
will
we
be.
should be able to the first quarter release publicly. We'll only be able to give data on 3 patients' muscle biopsies, which will be very limited, but at least it'll give us a sense of, you know, are we having a biological effect that we should be having, even though at 3 months we don't expect to see any functional improvements.
Mhm.
But it'll probably take some functional measures to then be able to have a thought about whether methylation could be a biomarker, which would then help us open up the door to measuring kids, etc.
And Amber, there's no placebo arm, right? And it's a 5-year study. But let's just say, God forbid, it does methylation, it doesn't pan out. And people are in this study, right? And
Mm.
they're kind of committed to 5 years, and avidity, say, gets over the line, or another siRNA gets over the line. Can those folks that participate in an AAV trial or epigenetic trial like yours, can they still take participate or take a treatment that is it like a siRNA treatment?
Well,
Or,
two
or
things.
maybe like a cocktail if both
Two
work?
things. One is, even if we did get approved and it worked, our mechanism is so different that it's not a bad idea. I mean, I would probably say take both. If Avidity makes it across the finish line, I'd say take both. One of the challenges, the other thing is Avidity's could work in the near term where ours may be better over time. And the reason I say that is the following. When you dose chronically every 6 weeks, the first time you dose, you're not going to get all of the cells. So let's say you get 70% of the cells. Those 70% of the cells are protected, DUX4 isn't coming out. The other 30% are at risk of DUX4 leaking out and dying. 6 weeks later, when that drug's out, well, not the full drug's out of the system, but you have to get redosed to keep enough level of drug in your body, it gets a different 70%. Some overlaps, but some doesn't. So now you have a different set of muscle cells that are at risk of getting DUX4. So the denominator of the number of cells you have to protect kind of goes down over time. Where one of the advantages of doing a one and done treatment with this disease is that once you protect that cell, it's protected. I mean, you are not going to be expressing DUX4. Let's just say those 70% cells, so maybe the other 30% are at risk over time, but at least those initial 70% are now safe. So that's why if I was thinking as a family, it was me and they were both on the market, I'd say use both, because maybe the cells that didn't get protected by Epi321, Avidity's thing may protect them somewhat. So I think they could be used together. Now take the scenario, it doesn't work. And it turns out that Epi321 was ineffective. if Avidity makes it to market, you can absolutely take that drug. When it comes to being on their trial, it depends on how Avidity wants to play. I mean, there's different ways to do drug development. You know, of course, they don't want to confound their data with other information, because what if it just works slowly and then they ended up on placebo? It could hurt their results. So part of it depends on how much we know how much you know about what it did. But I mean, they should be, if everything goes well, I mean, they're moving pretty quickly. They should be on the market so that if Epi 321, whether it works or doesn't work, you should be able to get Avidity's drug just buying it. I mean, you know.
Right, and that was my question. If 321 doesn't prove effective, you can, and you're in the trial, you can still go get on, and Avidity is a treatment, you can still get that treatment. I just
Oh,
wanted to
yeah.
make that clear and ask.
Yeah, yeah,
Yeah,
yeah, I would.
all right. We have about 7 minutes. Anybody else have any other questions? That I can ask questions all day long, but does anybody else have any other questions or comments that they would like to ask Amber?
Yeah, Tony?
Yeah, hi Amber, how are you?
Good.
Fantastic to see what you're doing. It is absolutely giving us a bit of a breath across the community. Keep doing whatever you're doing. I'm sorry, I'm not sure if anyone has asked that question before. I'm a little bit kind of troubled with a couple of things that I have listened to from Peter, and I'm sure you've seen the video of his in the last probably a week or so that he has released about understanding and giving us the ability to understand exactly what the drug is going to do. Have you been across that video and some of the questions that he has requested at the end of his video?
No, if you have
No.
questions, I'm happy to address them.
Okay,
We're very we
I understand.
basically work with anybody who will work with us. We
I
present the
understand.
data at
I
FSHD.
mean, look, your approach is very technical, and for us, normal, kind of humans in a way, we're not scientists. It's above us. Most of us, we just want to know if does it work or it doesn't work. So, Peter has done a brilliant video, and giving us a bit of a background of what he believes, from and translating it to our language in the last couple of weeks, and he came up with some questions at the end of his video. It is vital for me to understand if somebody from your team can actually answer those questions. That would be absolutely fantastic, because either he, didn't understand some of the things that you were translating, or we kind of took it in the wrong way. But there are vital questions which I would definitely love to get some answers to now that I understand a little bit about the drug.
So a couple things, Tony, that I would add.
Yes.
We've always tried to engage. I mean, I actually reached out to him when we first started working on FSHD, but he
Yep.
said he couldn't work with us because there was a conflict. So he's never talked to us about what we have. However, the only data he has at his disposal to analyze are like 2 posters and 1 talk at HGCT.
True.
So
True.
I'm sure it's really limited his ability to know what we're
Yep.
doing.
Of course.
Like I said, we submitted to the FDA thousands and thousands of pages. Like nowhere ever did we publish our 6-month monkey study as an example. So I would imagine, like if it was me looking at those 2 posters and the talk that my team gave, I would have tons of questions because it's like a little tidbit of a huge story.
Yep.
So
Yep.
I'm sure he's got a lot of questions.
Not a lot of questions, but
Yeah.
there are definitely there. From what I understand, they are manageable, and I would love to know, and I'm pretty sure Peter would love to know some of those questions. And again, because of the information that is available, but it's vital for us to kind of understand it a little bit more. If you think you can see that video and maybe put some of your technical people to answer some of those questions, will be fantastic for us.
I would imagine that he has the question about Cas9, because most people we talk to, like investors who look at their data, they're like, how do you get around the problem of Cas9 being
Yeah, one of them was that. Yes, correct. Yeah.
And we're like, we don't use Cas9, we use Casonics. So I would imagine he would ask about that.
Yeah, there's a few other questions in there, but I would definitely like to know some of the answers myself if I can, because I really want to see that drug going forward. And I really want to kind of get some, you know, results out of it too. To want to see, everybody wants to see some results, and it's fantastic what you're doing. You know,
Tony,
so
I
quickly.
think she's answered most of those questions already, and this will
Uh,
be recorded. So
okay,
we go
okay,
back and watch it, kind of bump it up
all
against
right.
her because I
I
still
came
know what
through
we're talking
halfway
about,
down,
but
so
I think
okay.
she's answered most of them.
Okay, I came halfway through, so
Yeah.
that's why I kind of didn't listen to the whole lot, and I'm sorry about
I
that. But
think
I will
just
go
her
through it.
explanation kind of went without,
Okay,
I mean, I
okay,
think it kind of touched on most of
all
them.
right.
Anybody
We'll
else
do that.
have
Thank you.
Yeah, hi. I do again. A much lighter and easier question, and maybe a little happier. Your sister is Rachel Salzman,
Yeah.
and so she's working for like you're both trying to work for a cure. So yeah, I just find that, you know, I have my daughters are sisters. So, you know, I hope one day my sisters can meet you, you know, the two of you, and that you guys are the answer to their prayers, you know.
Look,
So
it takes everyone. I mean, I'm seeing a picture of your daughters in the back, and they're probably so sweet. And
thank you.
hopefully they're always there for each other. And by the way, I always say when the kids fight, that just means they're engaging. It's better than if they're ignoring each other.
Mhm.
Are there any other questions? I want to, again, I want to be respectful of your time, Amber. I know you're very,
Any
very
questions?
busy.
Seriously, I mean, this is what you know, me and the team, we're so committed and thinking of the patients. You know, my family is asking me all the time, just given that, you know, unfortunately, one of my, you know, when we talk about lifespan, I just get annoyed when they say it doesn't impact lifespan, because dying at 60, that's not normal. And one of my cousin-in-laws died at 60, the other one's in a wheelchair, and then, you know, as you know, through the generations get worse. So I just get annoyed. I mean, I did lose it on somebody one time when they said it's not such a bad disease, and I was like,
We have members who've died as 16.
yes,
Another set
yes.
of sisters. Yeah,
Yeah. And I said, nobody's going to tell me that the kid's life, that if you see symptoms as a kid, their lifespan's the same. Nobody, I said, so they're like, well, yeah, most people, it starts in as an adult. I said, well, my cousin-in-laws, their symptoms started in their early 20s. You don't die at 60. I'm sorry.
Exactly. And again, I want to thank you for carrying that torch for us, because we shouted a lot, and sometimes people don't want to listen to us, but we appreciate that you're carrying that torch for us.
We're very driven. And by the way, I just the fact that you guys are here on a call at night, you are doing so much, and it really is. It's getting awareness. It's pushing people to do things. It's getting people to say, yeah, there's challenges, but we're just going to push beyond it and figure out how to fix them.
Well, thank you very much, and our condolences for the loss in your family with Oliver. I know that's tough and it never goes away, and we appreciate you fighting for our kids and our family members, and we appreciate you coming here and talking to us, because like you said, nobody else does, and you've always been very generous with your time with me and the parent group, and so I want to thank you, and we will keep working together, and if anybody has questions, you can email me. And then if it's okay, Amber, I can forward them to you and we can get those answered.
Anybody who's on this call, reach out to me if I can ever be helpful. I'd love to help.
Okay, thank you so much. And enjoy Boston and enjoy your daughter. And thank you so much.
Thank you all for everything that you do as well. And you know, just hugs to all the families. So thank you. Thanks for having me. Good night.
Good night.
Thank you.
Thank
Good
you.
night.